This is Paige, Gould, Nicole, Klitchko, Gracelyn, Marsh, and Yuventi presenting vitiligo. We're going to be discussing some of the general characteristics of the disease, the history, different diagnoses, as well as a stronger focus on the immune components of the disease.
Vitiligo is characterized by a patchy loss of skin color or depigmentation that can occur anywhere on the body, and it can also include the whitening or graying of hair and changes in the retina.
Typically, vitiligo is diagnosed through a visual examination in which the physician observes discolored patches in the individual and determines they meet the criteria of the condition. In some cases, typically in those where there might be evidence of another disease that could conflict the diagnosis, or the patient has lighter skin where the discolored patches are harder to observe, a skin biopsy may be done to observe the histology of the skin. In this test, the physician is looking for the distribution of melanocytes in the epidermis. In this slide are two photos of epidermis tissue. In the first image is a histology of a patient with vitiligo. In the slide, there is a clear absence of melanocytes in the stratum granulosum, though the destruction of the melanocytes occurs in the stratum basale. The tissue sample is taken from a region of depigmentation. Other areas of the body may still have typical levels of melanocytes. In the second photo is a typical histology of either someone without vitiligo or a non-pigmented area of a vitiligo patient's skin. The dark ridges is the melanin-producing melanocytes producing the pigment in the skin. Vitiligo is not exclusive to one particular ethnic background, though it is most prevalent in those with African descent simply because it is most noticeable on individuals with more pigmented skin. Vitiligo presents in both the depigmentation of the skin, as shown on all people below, but it can also present in depigmentation of the hair, which aids in the diagnosis of those with lighter skin. In Caucasians, the patches of skin that are depigmented appear almost pink due to the lack of melanin, allowing the pink tint of blood vessels and underlying tissues to become more prominent. In people of African and Hispanic descent, the condition presents in whitening of the skin, which is typically symmetrical and could lead to total loss of pigmentation in the face, arms, and hands. In some Asian ethnicities, the presentation can be more similar to those of Caucasians, with a depigmentation and reddening of areas of the skin, or it could present more similar to those of African and Hispanic ethnicities. It depends on how pigmented the skin is to begin with. To very basically give an idea of what causes vitiligo, there are three main schools of thought. The first two are interconnected, in which there is a genetic mutation that causes the autoimmune response to attack the melanocytes. The first type of development is an individual mutation in which the mutation occurred in the embryo and has not been present in the genetic line previously. The second type of development is that the condition is heritable, passing from parent to child as the dominant allele, in which only one copy is necessary for the trait to be displayed. The condition has penetrance, however, in which not every person with the genotype will equally display the phenotype. There are factors that can trigger expression and lead to depigmentation. Vitiligo is present in roughly 1 to 2% of the world's population, with approximately 30% of all cases to have a hereditary link. 60% of those with vitiligo will display symptoms before they are 20 years old, though the disease occurs equally in males and females. Vitiligo can onset at any point of an individual's life, though the majority are before the age of 20 and between the ages of 10 and 20. The earliest vitiligo development was immediately after birth, and often the onset in older individuals was brought about by some type of stress, whether emotional or physical, that resulted in the expression of the autoimmune disease. However, those that develop the condition in the formative years of their adolescence, such as between the ages of 10 and 20, report a higher percentage of bullying and low self-esteem.
The current model for vitiligo pathogenesis is not fully explainable, but the stresses that can cause for vitiligo can be traced back to certain stresses to the melanocytes. It can bring about oxidative stress that causes for an interruption of protein maturation in the endoplasmic reticulum, which leads to the activation and expression of UPR, or unfolded protein response chemokines, that call for immune components to the skin, ultimately targeting melanocytes to be destroyed. Next are components concerning melanocytes that can cause what is seen in the presentation of vitiligo.
HSP70I, also known as heat shock protein 70I, is normally secreted when microbes are able to damage cells. So this is a type of signal that is put out by a cell when it is under some kind of stress by an outside force. However, in vitiligo patients, we see this same heat shock protein being secreted by the melanocytes in the skin. And this usually occurs when that portion of the skin is under some kind of stress. And so what this does, this heat shock protein, it's going to activate dendritic cells. And so the dendritic cells, as we know, are what ends up going on to activate the T cells to induce a deeper immune response. So for the dendritic cells, these heat shock proteins are also going to present damaged pieces of the melanocytes to the dendritic cells. And this is going to activate the dendritic cells to go on and then activate the CD4 as well as the CD8 T cells. So with the cytotoxic T cells, the CD8 ones, you're going to have perforins and granzymes released from the cell, and that's going to induce apoptosis in the melanocytes. And then for the CD4 T cells, they're going to help B cells to create antibodies for the melanocytes. And this is also seen commonly in vitiligo patients where they have an increased number of melanocyte antibodies circulating within them, and this is thought to be where they come from. So now let's just take a closer look at the T cells that are surrounding the depigmentation portions of vitiligo. So there are found to be T cells that are specific for the melanocytes, and these are going to have an increased activation near the border or the perilesional region of the depigmentation. And in order to form an immune response against the pigment, they're going to need a melanocyte antigen presented to them, such as on MHC1. And so once this happens, they're going to secrete perforin in order to create pores in those melanocyte cells. And they're also going to produce granzymes, which will then enter through those pores created by the perforin, and they are going to induce apoptosis in those cells. And there are also found to be resident memory T cells in these areas after the initial immune response has ended. And so the CD8 T resident memory cells are what are most common. And what these do is whenever they're presented again with the melanin or the melanocytes, they're going to be able to respond much faster and create an immune response at a quicker pace because they have already been presented with them in the past. And this explains why repigmentation is not possible for these patients, because as soon as you go to give them the pigment again, they're just going to have those memory cells come and then fight and combat against the pigment again. So it's just going to keep a cycle of not being able to produce that pigment even if it's put back in your skin. NALP1 is also an innate immune system component, and it's found on the short arm of chromosome 17. And it's going to produce what is known as NACHT leucine-rich repeat protein 1. And this is found to be an innate immune system regulator, and it is found higher in immune cells as compared to other cells found throughout the body. And what this does is it produces inflammation and apoptosis in the cells where it is secreted near. Another thing it does is create the NALP1 inflammasome, and this is built by caspase 1 and 5 as well as the adaptor protein ASC. And what this inflammasome does is it increases the levels of cytokine interleukin 1 beta. And we know that these cytokines, when they are in a certain area, they are going to attract other immune cells to come because they're a signal that there is some kind of damage or stress in that particular area. And this also happens with inflammation as well. So it's going to be pulling in the activity of other immune cells. And so we have the combination of the inflammation from the leucine-rich repeat protein 1 as well as NALP1, and you're going to have an exacerbated issue with dendritic cells, because this inflammation is going to keep pulling dendritic cells into that specific area with the melanocytes. And then, as we saw with the dendritic cells before, they're going to be presented again with even more damaged melanocyte proteins, and it's going to activate more T cell receptors and B cells and antibodies, and it's just going to keep cycling like that as more and more inflammation occurs and as more immune cells are pulled into that area to damage the melanocytes.